Welcome to the fascinating world of cell biology. In this lecture, we will explore the intricate processes involved in the development, regulation, and function of different blood cell types. Understanding the biology of blood cells is crucial for diagnosing and treating blood disorders and gaining insights into fundamental cellular processes. Let's begin with the maintenance and self-renewal of hematopoietic stem cells. Hematopoietic stem cells, HSCs, are the building blocks of all blood cell types. HSCs possess the unique ability to renew themselves while also giving rise to different types of blood cells. This balance is controlled by specific signaling pathways and the microenvironment around the cells, which provide necessary support for survival and growth. This complex process ensures the continuous production of mature blood cells throughout life. Erythropoiesis is the process of making red blood cells. Signal pathways control the steps in making red blood cells. Hypoxia triggers the EPO pathway, which makes cells turn into red blood cells. Certain genes control making specific proteins needed to make red blood cells. Problems with these pathways can cause anemia or blood cancers. Granulocytes, including neutrophils, eosinophils, and basophils, play pivotal roles in the immune response against pathogens. The tightly regulated process of granulopoiesis involves successive stages of differentiation, maturation, and activation. Signal transduction pathways initiated by cytokines, such as granulocyte colony stimulating factor, GCSF, and interleukins, drive the commitment of multipotent progenitors towards the granulocytic lineage. Transcription factors, including C/EBP-alpha, PU.1, and GATA2, coordinate the expression of genes involved in granulocyte maturation and immune defense. Dysregulation of granulocyte differentiation can result in severe immunodeficiency or hyperresponsiveness. Lymphocytes, such as B and T cells, play a vital role in adaptive immunity. Their development involves complex processes like gene rearrangement and tolerance induction. B cells develop in the bone marrow through stages of gene recombination, while T cells mature in the thymus through selection mechanisms. Upon encountering antigens, lymphocytes activate receptors, triggering immune responses against pathogens. Megakaryocytes are specialized cells responsible for platelet production, essential for hemostasis and wound healing. Megakaryopoiesis involves endomitosis, where nuclear DNA replication occurs without cytokinesis, resulting in the generation of giant polyploid cells. Specific signaling pathways, such as the thrombopoietin, TPO/MPL pathway, regulate megakaryocyte development, maturation, and proplatelet formation. Proplatelets undergo fragmentation in the bone marrow, releasing platelets into the bloodstream. Platelets, small enucleate cells, possess unique features enabling adherence, aggregation, and the initiation of clotting cascades to prevent excessive bleeding. In this section, we will discuss the importance of red blood cells, RBCs, in oxygen sensing and the production of erythropoietin, EPO. RBCs are responsible for transporting oxygen and have specialized proteins, like hemoglobin, that bind and carry oxygen. They also have the ability to sense changes in oxygen levels and respond by increasing EPO production. This knowledge is essential for managing conditions related to low oxygen levels, such as anemia and hypoxia. During inflammation, immune cells, including neutrophils, monocytes, and lymphocytes, must efficiently navigate through the bloodstream and extravasate into tissues to combat threats. This process, known as immune cell trafficking, involves a series of sequential steps, including rolling, adhesion, and transmigration. Various adhesion molecules, such as selectins, integrins, and chemokine receptors, mediate the interactions between immune cells and endothelial cells lining blood vessels. Dysregulation of immune cell trafficking can contribute to immunodeficiency or chronic inflammatory conditions. In this section, we will explore the fascinating role of natural killer, NK, cells in our immune system. NK cells are responsible for identifying and destroying infected or cancerous cells. They possess specialized receptors and release potent substances to eliminate target cells. Understanding NK cell biology is crucial for developing effective immunotherapies against cancer and infections. Platelets are essential for hemostasis, preventing excessive bleeding upon vascular injury. When endothelial cells are damaged, platelets undergo a complex process of activation, adhesion, and aggregation to form a plug at the site of injury. Signaling pathways, including those initiated by thrombin, collagen, and von Willebrand factor, trigger platelet activation. This results in conformational changes in surface receptors, integrin activation, and the release of granules containing platelet agonists. The concerted actions of these mechanisms ensure the rapid formation of a stable clot, sealing the damaged vessel. Blood cell disorders encompass a wide range of genetic and acquired conditions affecting various aspects of blood cell development and function. Thalassemias, for example, are a group of inherited disorders characterized by abnormal hemoglobin production, leading to severe anemia. Myeloproliferative disorders result from dysregulated proliferation and differentiation of blood cell precursors, leading to abnormal cell counts and increased risk of transformation to leukemia. Understanding the molecular basis of these disorders enables the development of targeted therapies and improved diagnostics. The endothelium lining blood vessels plays a crucial role in maintaining vascular homeostasis and regulating interactions with blood cells. Endothelial cells express adhesion molecules and produce cytokines that influence the behavior of circulating blood cells. Blood cells, in turn, can modulate the endothelium's function through the release of soluble factors. Dysfunction in the interplay between blood cells and endothelial cells can contribute to thrombosis, inflammation, and cardiovascular diseases. Studying these interactions helps elucidate mechanisms underlying vascular disorders and identify potential therapeutic targets. Hematopoietic stem cell transplantation, HSCT, offers a curative treatment option for various blood disorders, including leukemias, lymphomas, and inherited bone marrow failures. HSCT involves the infusion of healthy donor-derived HSCs, which subsequently repopulate the recipient's bone marrow, re-establishing normal blood cell production. Allogeneic HSCT, where HSCs are obtained from a compatible donor, can also have immune-mediated effects, known as the graft versus leukemia effect. Recent advances, such as reduced intensity conditioning regimens and targeted manipulation of donor cells, have improved transplant outcomes and expanded the therapeutic potential of HSCT. As medical professionals, it is crucial to comprehend the complex and detailed world of blood cell biology. This understanding is essential for diagnosing, treating, and preventing blood disorders. Ongoing research in cell biology will continue to provide valuable insights and improve patient outcomes.